Hello everyone, my name is Bongsung Bae. I'm a PhD student of the Gwangju Institute of Science and Technology. I'm here to talk about our current research called LOGICS, a framework for learning optimal generative distribution iteratively for the focused chemical structures. This is the table of contents for today's talk. Let's jump into the introduction. The recent trend in de novo drug design is the introduction of deep generative modeling. The interesting application of generative modeling is to generate the potentially bioactive molecules to a protein target. But we usually face the problem that we don't have enough data for a specific protein. If there's a lack of data, the first thing you would consider is the transfer learning. In transfer learning, we transfer knowledge of a model to a model for domain A with big data to the model for domain B with small data. In this study, we are interested in a special type of transfer learning approaches called generator-predictor collaboration, or simply GPC. The terminology GPC is created to explain the concept of this study. The figure on this slide describes the GPC scheme. It has the pre-training phase where we pre-train the generator and construct the predictor, and fine-tuning phase where we bias the generator towards the desired properties. The GPC approach has several characteristics. First, the fine-tuning dataset is not provided. Second, the fine-tuning data is generated by the generator. Third, the generated samples are evaluated by the predictor, and these predicted scores and samples are used to fine-tune the generator. These four previous studies listed on the bottom have proposed a method that perfectly fit under the GPC. Something we have observed when we used GPC is that naively training the generator to focus on high-score samples usually causes the mode collapse of the generative distribution. Mode collapse can occur when the generator only receives the signals from a single mode among the diverse target distributions. The previous studies were not able to detect this problem because the model evaluation is mainly performed by the predicted scores, and only checking the diversity of generation doesn't guarantee the discovery of different target modes. Thus, our research goal is to propose a novel strategy for boosting the exploration of target space and evaluate the model on the distance between the target and generative distributions. In terms of data sets, we brought the pre-training data from Guacamole benchmark, and for the predictor training, we used the pre-processed data set from the diverse DRL paper for KOR bioactivity optimization case, and we directly downloaded the bioassay data from the PubChem for PIK3CA case. For predictor module, we used the random forest regressor with Morgan fingerprints. The 5-fold random cross validation is done, and the best performing predictor is selected based on the R-squared score. So let me explain about this proposed framework. We first pre-trained the prior generator G theta with the 1.6 million generic molecules. As you see here, the architecture of the generator is three-layer SDMs, and it is trained to be a smart language model. The fine-tuning phase of the framework starts with the initialization. The bioactivity predictor is trained. The agent generator G5 is initialized with the prior generator's weights, and the experience memory Q is constructed with the 100,000 generations from the prior. The iteration starts with the sampling from the memory Q and the agent generator. The sample molecules form the initial Tolman candidates, and these candidates go through the three-stage Tolman selections. The first stage selects the molecules with higher predicted bioactivity. This is the necessary step since we want the agent to become the generator of higher bioactivity. And the second stage selects the molecules with lower agent likelihood. This is for boosting the exploration ability by obtaining the molecules that are not likely to be sampled by the agent. The third stage selects the molecules with higher prior likelihood. This stage is purely for the regularization because we want to prevent the catastrophic forgetting of the generator. At the end of the iteration, the finally selected molecules are used to update memory queue and fine-tune the agent G5. That is the end of one iteration, and repeat this process until the predefined number of epochs met. Now let's talk about the results. So this is the result of PIK3CA bioactivity optimization. We compared our proposed method to the vanilla GPC model and the 3 other methods from the related studies. FCD here is the Fresher cabinet distance, and OTD is the optimal transport distance. These are the metrics that evaluate the distance between the generative distribution and target active distribution. In this table, you can see the distances are minimal when we use the proposed method. We also performed the TSNE transformation of Morgan fingerprint vectors from the generations and the known target actives. The blue colors are the generations from each model, and the red colors are the known target actives. In the perspective of distributional learning, the proposed LOGICS has shown the closest generative distribution to the targets. The experiment on KUR bioactivity optimization also showed the similar result to this experiment. For conclusion, we developed a novel framework LOGICS for generating target protein active molecules. And the proposed method showed the better performance in terms of learning closer generative distribution to the known targets, actives. This work was supported by the National Research Foundation of Korea, grant funded by the Korean government. Me and the Professor Ho Jung-nam have contributed to the research, and I want to give special thanks to my colleagues in Bioinformatics and Intelligence Lab. These are the references of today's talk, and that's it. Thanks for listening.